Welcome everyone. My name is Sandrine Heitz and I'm the LCN co-director at Imperial. It's a great pleasure to introduce two speakers today who will tell us about some new facilities at Imperial College that are linked with the LCN and will support LCN research. So we've had a range of different facilities talks that are posted on the LCN YouTube channel. So I will send that link again. There were about 5 minutes each because there were sort of well-established facilities, but here we've got some two brand new shining facilities. So we thought we'll have 15 minutes to let our two speakers introduce them. So it's a bit more detailed information. So we'll kick off with Alberto Colauto. So a little intro for Alberto. He did his PhD in chemical sciences at the University of Padova. Then he did postdocs at the Weizmann Institute and at Goethe University in Frankfurt am Main, where he applied advanced EPR techniques related to biological and spin probing techniques. He joined Imperial College in this spring and is managing a brand new facility, Pepper, and I will let him get started on his presentation. The floor is yours, Alberto. Thank you very much for joining us today.
Thank you very much for your very kind introduction. So today, to introduce you to the Pepper facility, I decided just to showcase what are the capabilities of Pulse EPR by showing examples ranging from biological systems to solar materials. And well, definitely a disclaimer, I'm not going to discuss all the possibilities of Pulse EPR, also in interest of time, because there are entire techniques that I will actually not be even able to mention. So maybe the first question to start with, just a second, perfect, is what can be measured with Pulse EPR? What is Pulse EPR good for? And I try to make a list of what CWPR is most suitable for and what Pulse EPR is most suitable for. And as you can see from the list, there are definitely certain applications where CWPR is most suitable. I would say the main application of Pulse EPR is when one has interactions that are smaller than the inhomogeneous line width of the sample. This could be, for example, hyperfine couplings, small hyperfine couplings. By small, I mean on the range of megahertz or even smaller than that, that can report on electronuclear distances or spin delocalization, so basically giving us the shape of our semi-occupied molecular orbital. On the nuclear quadrupolar interaction, environment of the nucleus, bonding geometry, weak electron-electron dipolar couplings, which can be translated into electron-electron distances, very relevant in structural biology. I will actually show plenty of examples on this. Or also electron spin relaxation times, both T1 and T2, and the identification of the spin state S of the system under investigation. So, having to choose which example to pick, they will be mainly focused on biophysics because this one is actually my background of my last two postdocs. But I will also cover something about carbon nanostructures and one example about sustainable energy. And now, starting with the biophysics, I will start talking about distance measurements in biological system, electron-electron distance measurement. This will actually be the only time in which I will show a pulse sequence. I'm doing it now because that's a fairly easy one. It is called diapulse sequence, and again, it measures the electron-electron dipolar coupling, and through analysis of the measured trace, one obtains a distance probability distribution. That's actually important. There is not just information in the location of the maximum, but also in the width of the distribution itself. Measurements have to be performed on frozen samples, typically 50 Kelvin for the nitroxide radicals or aminoxyl radicals in general used for this kind of applications, and can be used to report on distances in the 2 to 8 nanometer range. As mentioned, the analysis of these traces is at least in this approximation parameter free. And I will start from this example here, structure and dynamics of nucleic acids. Now, what you see here is a double-stranded DNA, a short double-stranded DNA. Well, DNA itself is diamagnetic, so it cannot be investigated with pulse CPR spectroscopy that looks at paramagnetic species, radicals. We need to put our reporters. In this case, it is this modified cytidine called cisspin. The cisspin actually carries the nitroxide center. It is the spin-bearing moiety. If you look at the structure, this system here is entirely rigid. Just look at whichever angle you want. Either it is constrained by chemical bonds or by hydrogen bonds. But this actually means that the width of the distance distributions report on the width of the conformational space when the sample was frozen. Now, the samples are done by keeping one of the labeling position constant and just changing the other. It is a series of different samples that are labeled in pairwise positions. I think not surprisingly, going down the sequence, the distance become longer, but also there is a dependence of the flexibility. And this one can be modeled to give back information about the dynamics. In a very simple geometrical model, one sees, for example, that the experimental data are consistent with a stretching motion of the DNA itself. It is also possible to correlate the results of the measurements with the result from molecular dynamic simulations, especially performed with the newer amber force fields OL15 and BSC1. They're able to give a very, very nice reproduction of the experimental results, meaning we have a one-to-one match. It is really possible then to use the information from ND to learn something more about the behavior of these nucleic acids. The next example I'm showing you is now about a short double-stranded RNA, and in this case, this one has been internalized inside cells, in this specific instance, Xenopus levis oocytes, and it is only one labeling pair in this case. And what you see here, it is a reproducible change of the distance probability distribution when going from the solution to the intracellular environment. It has been done on several biological repeats and always reproducible. Now, this one could be actually modeled with the help of molecular dynamics. What we actually found out is that even such a structure, such a fairly rigid structure, undergoes compaction inside the intracellular environment. Now, this is really the kind of detail information that we can get out of these measurements. Completely changing system. We switch now to proteins, and that's the case of a diubiquitin, which is labeled with a rather popular MTSSL spin labeled or metantiosulfonate spin label. You can see the structure in the slide. And this one, well, it is definitely characterized by a high mobility. You can see here the chain that connects it to the backbone. What has been done here is to take a series of positions, labeling all of them, measuring the distances, and building a model that simulates all of them together. And what we can obtain here is not just the second position of the other ubiquitin. We assume that one is fixed. We treat them as rigid bodies. It's not just one position of the second one. It is a whole conformational space, including a concept of probability where the second ubiquitin could be found. And the same kind of argument can actually be done incubating the diubiquitin in the presence of binding enzymes. There are here two examples. This red one is a ubiquitin binding domain that leads to so-called conformational selection. You see, actually, the conformational space becomes more restricted, still stays within the one of the free system. Or if incubated with the deubiquitinating enzyme OTUB1, it even leads to population of certain parts of the conformational space that were not populated by the free system, leading to what is called conformational remodeling. Now, this one is my last example. This one was the last example about application of these electron-electron distances. It is also possible to obtain information about mechanism of biological reactions. And what you see here is a study and a comparison of two transporters. These transporters are ATPases. They get energy from the hydrolysis of ATP, and this one happens in the presence of magnesium as metal cofactor. Now, magnesium is diamagnetic. Again, we would have absolutely no signal, but what you're able to do is to replace magnesium with the paramagnetic manganese. This one doesn't alter significantly the kinetics of the system, but it is actually possible now with the paramagnetic manganese to probe the environment to get information from the coupling with the nuclei. You see, for example, here the phosphorus nuclei, which lead, for example, to the possibility of following the reaction kinetics. You see here a comparison between the two systems. The principle according to which it works is fairly straightforward. When ADP is bound, so the hydrolyzed form, only one phosphate interacts with manganese. When ATP is bound, so before hydrolysis, two phosphates are bound to the manganese. So we just expect a decrease of the signal when the hydrolysis reaction takes place, which is actually what we find. There is actually a peak in the middle that's actually related to, again, already to some different reaction mechanism of these two otherwise rather similar enzymes. It is also possible to incubate the system in the presence of vanadate. Now, what vanadate does is after the hydrolysis of ATP, it replaces the last phosphate, the one that has been hydrolyzed, but then it locks the system in, and the system is just not able to recycle it anymore. And here we really see a clear difference. For this ATPase here, which is called MSPA, both hydrolysis sites are active, and we see a very strong vanadium peak. Whereas in BMRCD, only one of the two sites is active. The other one is actually non-catalytically active. Our vanadium 51 signal is much weaker. We're actually getting insights about the mechanism of this biological reaction. We can even go further by doing experiments with 15 unlabeled ATP. Fast freezing, fast means with respect to the reaction time that we measured before with the phosphorus experiment. We see spectra that are completely different. And this one actually points out different coordination ways of the ATP to our proteins. What is now the next step? The next step is going to be performing pulse EPR spectroscopy by generating electrochemically paramagnetic states inside the EPR tube. This one is already possible for continuous wave. You see actually here the results from this publication, and this is now going to be ported to the pulse EPR, to the pulse EPR spectroscopy, to take advantage of all this amount of information that can be obtained from the pulse techniques. This one was for the biophysics. I will now switch completely. Carbon nanostructures, fullerenes. Here actually the question was, one takes a reduced fulleropyrrolidine and one puts a hydrogen molecule inside. A hydrogen molecule in its auto state, so it has an I equals to 1. The question is there actually coupling between the electron spin and the nuclear spin of the hydrogen? And our pulse EPR experiments actually show that yes, this coupling is present. And this measure was actually attempted with other ways, but it was always elusive. And here there is actually a very clear confirmation in this case. Or another example, I find this really neat. It is again a fullerene that is substituted with two nitroxides. Once again, this spin 1/2 species. Now, taking the system like this, the spin 1/2 species are uncoupled to each other. If we do an experiment that correlate the nutation frequency with which through the formula that I wrote at the bottom of the slide is actually correlated to the spin state, we just see one line corresponding to spin 1/2. It just means the two species are not correlated to each other. In the moment in which we shine light, we can induce a triplet state on the fullerene, the whole system becomes coupled. And we can actually see clearly the signature of the quintet state, and S equal 2. Not even just a signature, we can even get the individual spectra. And this one will lead to my last examples. Application to the solar materials, which actually comes from the LCN, comes from Imperial. And it is assigning the spin states in singlet fission materials. And you see here a spectrum that was obtained. This one is a pentacene. the system can evolve through a quintet state. The question is, which one of these peaks are assigned to the quintet state, which one to a triplet state? Repeating the same technique, we can clearly identify and clearly see the signature of this quintet state. So I'm done with the examples. Just shortly about the access to the facility. Feel free to approach me or Maxi. We also have a website. A disclaimer about that: it is still work in progress. Hopefully by the end of the year, beginning of next year, we should have a more definite version. We also have a form to request access to the facility. There is a mailing list. I plan to send out the first issue of this mailing list by the end of this year. So I would invite you to subscribe to hear news about our facility, how things are going, what is the equipment that we have. And well, thank you very much for your attention.
Thank you very much, Alberto, for this great presentation. We will keep questions to the end of both presentations, if that's okay, unless someone's got some burning questions now. Feel free to put them in the chat or raise your hands at the end of the talk. No, I hear silence. Okay, let's move on to James, and then we will move on to questions after that. So James Douglas is the facilities manager for the Imperial Center for Cryo Microscopy of Materials. Sorry, ICM². He studied the physics at the University of Bath, and then did a very LCN appropriate Masters in Nanoscience to Nanotechnology at Swansea. He then moved off to Oxford, where he did his PhD in materials and started working on atom probe tomography. And he honed this skill during his postdoc and also as an atom probe staff scientist. So he also joined us this spring, and we're very pleased to hear you today. Thank you very much, James, for your presentation.
Thanks, Sandrine, for that introduction. Thank you very much. So yes, I'm James. I'm the facilities research facilities manager, as Sandrine said, and I'm responsible for the atom probe tomography part of the ICM². We were very pleased with the parentheses. We were very pleased. So what I'm going to do today is go through a bit about the facility, where it currently stands, as it's still being installed, still getting to grips with everything. And this is also being recorded, of course. So if you are intending to carry out atom probe or use the facility at some point, this can be a bit of a record to be able to look through and go, what do I need to think about before applying for use of it? What's the requirements? What information can I get out of it? And I'm going to go a little bit into the sample preparation, because the atom probe analysis and sample preparation are so intrinsically linked. So here's a click. There we go. So just a very little bit of a background. People are more and more interested in cryo sample preparation, sample transfer, and then cryo analysis for EM and also for atom probe. And this is because it allows us to analyze things we previously couldn't analyze, like frozen liquids, liquid-solid interfaces, and especially like light mobile elements, hydrogen, deuterium, lithium, things like that. To be able to do this sort of thing, you need to be able to make samples also at cryo, and that's cryo-focused ion beam that's traditionally used for that, or not traditionally, is starting to be used for that. And it's challenging, but it's starting to become viable and comparable to conventional FIB lift-out. And the point of this facility is to be able to integrate multiple different microscopy techniques at cryo, at vacuum, to be able to transfer between them and be able to look at the same material, ideally the same exact sample, with different techniques to give us a better understanding of what's going on in the structure and the composition across multiple different length scales. And the way we do that is we link up our atom probe, our focused ion beam, and our TEM with an inert atmosphere glove box and transfer samples between them while controlling the environment in terms of atmosphere and also temperature. So to do this, it's quite tricky, and there's a lot of logistics involved. So this is part of why I wanted to have this as a thing of record, to be able to go have a big think about exactly how we do all the sections. And this is one of the facilities trying to do, is develop these protocols to be able to apply cryomicroscopy to lots of different. Oh, I've had a thing in the chat to be able to speak more slowly. Very well. The facility is designed to promote and develop the techniques and develop the processes to transfer between each of the different microscope techniques and be able to develop them for these environmentally sensitive materials. So at the moment, we have 2 instruments installed. We have the state-of-the-art atom probe system, the CAMECA LEAP 5000. This is equipped with a laser, so we can also analyze non-metallic materials, so non-conductive materials. It has relatively high detection efficiency and has high mass resolving power, capable of isotopic analysis. And this is in the Royal School of Mines. We've also got installed the plasma FIB, the focused ion beam, that again is state-of-the-art. Unlike the gallium ions, the gallium focused ion beams that have been around for quite a while now, these are multi-ion source instruments. So we have xenon, nitrogen, oxygen, and argon with up to 2.5 microamps of current for high throughput milling. And we also have a cryo stage, which allows full rotation and tilt, and we can change that to our standard stage when we want to. It's not particularly difficult. And we also have a cryo manipulator for lift out for TEM and for atom probe, so we can keep everything at the right temperature whilst we're making our samples. Again, this is across the hall in the Royal School of Mines. These are linked together by the Ferrovac high vacuum cryo transfer module. It's a little suitcase that is attached onto both machines that we can use to extract samples and sample mounts and place it from one into the other, and also the glove box when it arrives. And the glove box and the high resolution TEM should be delivered and installed later in 2022. So at the moment, we've got the atom probe and we've got the FIB. So a bit of a background on the atom probe, because up until now, only Oxford has had any modern atom probe systems in the last few decades or so. Sorry, is there anything else in the chat there? There we go. Yeah, so only Oxford has had them. So I wanted to go through a bit of the technique because many people may not be aware of it. It's a high resolution microscopy technique that gives us sub-nanometers up to sub-nanometer spatial resolution and ppm compositional information in a very small nanoscale volume. Oop, need to click on the screen. We need to have a needle-shaped specimens, so I'll go into the sample prep as well, with an apex less of 100 nanometers, cooled down to 50 Kelvin or so, and then we expose them to very high electric fields. These are then evaporated by a voltage or laser pulse, accelerated towards a detector, and we can use that time of flight and the XY location of the detected ions, combine it with the order of arrival onto the same detector elements, and we back project them. We make a 3D reconstructed model of the original specimen size and shape, and also we get information about each of the detected ions. So what we get the nice spatial resolution, we get 3D information, and we can detect all species equally, although there are a few caveats, hence me asking for people to get in contact before deciding to do atom probe. And here's where it sits in terms of the spatial resolution and the detection range in between the other techniques that typically use SEM, STEM, SIMS, EPMA, and things like this. So it fills a nice little niche in the center. And we can get up on the straight flight path instruments up to 80% of all the ions on the detector. We've got a separate system for that. We don't necessarily need calibrated standards, as the samples are all mostly unique in terms of the size and shape, because your sample is effectively the ion optic for this. So they're all relatively unique, but the standardization of analysis conditions and data reconstructions is important. And what it's really good for is nanoscale features where you have local changes in composition. And because it's such a small volume, as I'll show in a second, you need to have larger scale analytical techniques. You can't just jump into it and go, I'm going to look at this particular piece of material. You need to be able to understand more about the context of the very small sample volumes you're looking at. So normally we use focused ion beams, so high energy ions used to mill things out. So we'll make a cantilever of about 5 by 5 by 25 microns. We'll then pick that up like you would a TM lamella. We'll mount wedges of that onto individual support structures. These are some examples of some commercial silicon microposts. You put a little chunk of material on top of those. It takes about an hour per sample to make, and that's for relatively simple ones. So it's not a, and these are all, it's a destructive technique. So one sample means pretty much one analysis. So you can take a lot of instrument time to make your samples. So what we do is once we've got our nice wedge of material placed onto a support system, we can then, that allows us to sharpen this into a fine point. And at this point, we can see any sub-microstructure that might be within the sample. This is from a benitic steel, so we can see lots of little grains and laths and carbides and things. And then this is the sort of size and shape of the sample that we need at the end. But we'd only actually end up collecting data from, say, the top 50 nanometers of this sample. So the sample is, if you want to be able to look at a very particular feature, placing that feature right at the top is the difficult part. As I said, we've got a plasma FIB, so now we can actually do slightly different methodologies. This allows us to, instead of picking up a small piece of material and sharpening it, we can dig a very large small hole in the hundreds of micron scale, And that allows us to have an isolated sharp point in the center of our this little satellite dish. And this means you can have multiple samples made in a single substrate and just drill holes across it. However, you do have a bit of an issue with sample target specificity, and it does use up a lot of material as well, but it's quite a useful thing to do. Anyway, this is what the atom probe data looks like. You get a mass spectrum. So if you're similar to like TOF sims or anything like that, you get a time of flight mass spectrum, and that turns into mass to charge ratio. We get isotopic fingerprints, and then we get a 3D dot model, which is every single point on this is a single ion that has a defined mass to charge ratio, and then we can reconstruct the size and shape of this from ion projection models, and then you can also do things like TEM of the size and shape of the sample before and after you've analyzed it. And then once it's a 3D dot cloud, then you can then it's effectively you can link up dots, you can join the dots, how many dots of different types are close to each other, how far away they are, and you can isolate different regions of this. And this allows you to extract out nanoscale clusters or interfaces, which is the real power of atom probe. So in this example here, they're looking at in a like 4 billion year old zircon, looking at tiny lead clusters to get isotopic ratios of the lead to work out the age at which these clusters formed. So that's a really cool, really cool overview in that paper there. So we need to click on the screen again. Let's go back a little bit. There we go. Because atom probe samples are so thin, you can also do anything pretty much you can do with TEM, you can do on an atom probe sample, including STEM as well. So you can do diffraction analyses, you can do spectroscopy, and you can get really nice spatial information. And this is what we want to do with the facility. So you'd have your material, you'd make a atom probe sample, you put it into the high resolution TEM, get a better understanding of the structure, including all the dislocations, all the grain boundaries. You can do your misorientations with TKD and stuff like this. And then you can analyze in the atom probe to look what the compositional variation is linked to that. So here's on pearlite steel. And you can actually see in section F at the bottom there, the size and shape of the analyzed volume from atom probe compared to the TEM sample. We only actually get the core of the material. Cool. And it can also use to inform the reconstruction parameters. So cryo sample preparation is what we're interested in as well. So can we freeze stuff and then look at it in the TEM and the atom probe? On this left example here, this is a droplet of water on a rough surface of tungsten. So they're able to just freeze water on the end of that and turn that into an atom probe tip. And it's good for being able to do a look at samples sensitive to heating during FIB sample preparation. This includes things that have heat, hydrogen or lithium or something, where the mobile elements like to wander away during sample prep. So we can do it on a cryo stage as well. And but maintaining that cryo and vacuum environment for all your samples between different instruments is the key point of the facility. So this is, I'll skim through this quite quickly, so this is something people can look back on. The three main stages of doing atom probe is, can I put the piece of material that I'm interested in right at the very top of my apex of my tip? That can be very challenging, and you might need quite a while to work up your skill on the focused ion beam to do that. Are you analyzing it with the correct parameters to be able to make sure you're not inducing artifacts? So in this case here, this is just a quaternary quantum dot system that I did some work on last year. And then the final part is making sure that the reconstruction, the data analysis part, is actually physical. And then can we link it back to anything else we know about the material a priori to this? So we already had TM images of the quantum dot system and scanning and tunneling microscopy. And that allows us to look at in this case, the indium concentration of a quantum dot and be able to look at the size and the shape of this distribution compared to the other elements. So quick case study before I wrap up. As I said, the facility is relatively young, so this is just stuff I did in my previous position. So looking at composition of primary, secondary, and tertiary gamma prime precipitates in a nickel superalloy, you can see the size of the tertiaries down there. It's very difficult to isolate just the composition of just the tertiaries if you're using other techniques. Here we can do that in 3D, as you can see by the images on the top right. We can look at compositional variation across these interfaces, and we can see in this case that the hafnium across this primary gamma prime to gamma, the composition of the distribution of hafnium changes at a different rate compared to the other elements. And you can look in that paper to see exactly why that's interesting. But this is the sort of thing that we can see. And you can see the composition variation of hafnium on the right-hand side of that. You can see differences down to the 0.1 to 0.2 atomic percent. We can look at nanoscale solute clustering. In my previous position, I was looking at radiation-induced clustering in reactor pressure vessel steels. So we can look at the clustering of solutes like nickel, manganese, silicon, and phosphorus that are formed due to irradiation and be able to see these. You can't really see it with other techniques. And we're able to see homogeneous and heterogeneous nucleation and growth of these clusters and link that back into the back that into the mechanical properties. So again, this is something that you can look back on the recording. If you want to come and do some atom probe, please do get in contact, but think about what do you want to look at, size and shape of your samples you want to get out of, because we're looking at such small regions, we need to make sure it's representative. Are you air sensitive samples? Are they cryosensitive? All these things to look for process flow. What sort of composition do you expect to find and the variation in that? We can see everything, but with a few caveats. And we do service work as well. It depends whether training on the sample prep, data collection, or data analysis would be useful for whoever's doing it. But PhD students starting out, almost certainly. If it's a one-off to check something, maybe not. And please do get in contact. So my email address is up there, and the academic lead is Professor Finn Giuliani. So he's also someone to get in contact with. Yep, I think I'll wrap up there. Thank you.
Thank you very much, James, for a whirlwind tour through atom probe tomography and cryo EM. So the floor is now open for questions to both Alberto and James. I've just put in the link for the LCN recordings where you can the YouTube channel where you can find this information. And look back on it. If no one's got any questions, I will just kick off. I think what was really nice oh, there is a question. James, does your workflow also work for work proteins, i.e. cryo? Actually, my question was along those lines as well. It's really powerful that you can both work on cryo samples as well. And yeah.
In terms of
badly formulated question, James. Apologies.
Maxi's got a mute. All right, go for it.
Yeah, so I actually spoke to Finn when he was submitting the bid, so I'm a little bit familiar with what's going on, but only quite peripherally. But it sounds all potentially really powerful and very exciting also for what we want to do. But I guess I have so little understanding of all the techniques individually that you're trying to put together that I guess I'm not really at the stage of really being able to put one and one together to see whether you can study things like proteins in the cryo state with your atom probe, for example, or whether this is really geared to much higher concentrations that you have in typical material samples. So I'd be grateful if you could comment a bit more on that.
It depends what the actual protein is made out of and what the size of it is compared to the apex of the atom probe tip. And because it's like with any time of flight mass spec, if you have a large complex molecule, what you're actually detecting is it all shattered up into all the individual parts. So you might just see lots and lots of different carbon peaks and things.
Right. So let's say it's about 30 kilodaltons to 50 kilodaltons.
The maximum dalton we normally look at is about 200, 300.
Okay.
So you wouldn't, if you wanted to, it's just to do with the flight path. You wouldn't be able to see those sort of things. You'd be able to see things that would be, you just see a load of carbon, unfortunately. People have used it for looking at cells, because cells are actually on bacteria. You can look at individual walls and things, but I don't, as far as I'm aware, proteins are not necessarily possible at the moment for the atom probe part. However, for the cryo sectioning and things for the FIB and then the TEM, that's more plausible.
Okay.
So it's the sample preparation part of it is probably going to be more useful to you rather than the atom probe itself. However, it depends if there's something within the proteins that you're looking for. Like, is there a specific element that you'd be able to say, if it's there, that is good. If it's not there, like
molybdenum in this case. So
yeah. So if it was just a question of, I've made a sample, has it got any molybdenum in above a certain concentration? Potentially, you could say that, but the structure of it, I don't think would be retained in the data.
Okay, so we can't really tell how it's oriented on the surface or how that's.
I'd have to look at it, but as far as I'm aware, people have not been able to do that sort of thing. They've done thin films, so you can do thin films and work out what a known molecule would fracture into, and be able to see the different sections of it. But I think proteins are out of the capability, at least at the moment.
Okay, but it sounds like the other two would still be useful. So maybe we'll drop you a line anyway and see whether it would be great to hear your thoughts. If it's not doable, then fine. But otherwise, yeah, thanks. I don't want to take up all the question time, so thank you.
Just following on that, and you, if it was a dilute metal in a structure, I mean, Alberto mentioned that you can get distances between 2 and 8 nanometers between sort of spin centers. If the spin centers come from a metal, is this something that in APT could be resolved, or what's the resolution, spatial resolution in atom probe, like the minimum distance between two metals that you could resolve?
It's about in an idealized situation, it's about 0.2 nanometers is about the theoretical limit.
So actually,
Yeah.
they could work together, except that you can't get the big mass fragments. Okay, great. Sorry, I have a question in the chat for James again. What is the biggest challenge in preparing samples for APT?
Biggest challenge is, it all depends on your material system. It's mainly if you have a very specific thing that you want to analyze, making sure that that's in the top 50 nanometers of a 100 nanometer needle, and then it's within the top section of that, but not right at the very, very top, because otherwise it's been damaged by your sample prep. And that's the part that takes a large amount of the time and the skill. So in the example that I showed of the quantum dots, that took a reasonable amount of time to be able to get right, to be able to make sure you haven't destroyed your material you're looking at. In the case of the other example that I had with the nickel superalloy precipitates, that's relatively a homogeneous material. So you can just make a sample and then you will have the little tertiaries in there somewhere. And that's really quite easy. It's when you have a very, very specific thing like finding the apex of a crack tip. So just at the top 10 nanometers of that, that's really, really, really rather tricky. I've tried it a couple of times and I did not like doing that. Okay,
thank you. Any other questions? And related to that, a question that we often get for EPR techniques. So what are the sample requirements for EPR? Are there any restrictions for you, Alberto?
In terms of sample requirements, so we have definitely limits for what concerns the concentration. Lowest concentrations that we can detect can go as low as 10 micromolar, even possibly lower than that. Upper concentration, if it goes to pulse, it stays in the limit. I would say 1 millimolar. It is possible to detect. It is possible to do experiments, but it starts coming definitely at the price of rather fast relaxation. So it's more difficult to perform experiments. What just matters is not just the bulk concentration, it is the local concentration. So it should be always thought to have some cryoprotectant in order to make sure that the sample forms a nice glass. In alternative, it is possible to use very fast freezing techniques, as for example the rapid freeze quench. The one should still yield, even in the absence of cryoprotectant, rather good dispersion of the paramagnetic species. And in terms of volumes, if it goes to the higher frequency that we have, it is the Q band. It is possible to study as little as 5 microliters of samples. If it goes to the X band, we can go as little as 20 to 25 microliters. Those ones are the minimum requirements, I would say.
Wonderful. Okay, great. I don't see any further questions in the chat, but you've very kindly provided your contact details, so I'm sure we can get in touch with you for further questions. And as this is part of an LCN seminar, also please, participants or investigators from UCL and King's College London hopefully can join as well in these experiments. And so thank you very much, and we'll look forward to great experiments on atom probe and EPR. And thanks very much for your presentations and everyone for listening.
Thank you.
Bye.